Imagine for a moment that you are walking through the streets of the Western Netherlands in the winter of 1944. It is arguably one of the darkest moments in human history. The Second World War is raging, but here, the enemy is not just the occupying soldiers, it is the cold, and more importantly, it is the hunger. They called it the Hunger Winter. The German blockade had cut off food and fuel. People were desperate. We are talking about a population trying to survive on less than 30% of the calories a human being actually needs to function. It got so bad that people were grinding up tulip bulbs to make a sort of paste just to have something in their stomachs. They were burning their furniture to stay warm. It was a humanitarian catastrophe. But here's the thing that makes this moment pivotal for our understanding of biology. Amongst the survivors of this horror was a young girl, 16 years old, named Audrey Hepburn. Yes, the Hollywood icon. She survived by eating tulip bulbs. And while she went on to become a symbol of elegance, the shadow of that winter stayed with her physically for the rest of her life. But the story does not end with her generation. This is where it gets terrifying. Decades later, scientists looked at the babies born to women who were pregnant during that famine. They found that what those mothers ate, or rather, did not eat, changed their children forever. And not just their children, the grandchildren. The effects rippled down through time. We used to think our genes were written in stone, a script that could not be changed. But this story, the story of the Epigenetics Revolution, tells us that the script can be rewritten by the lives we lead.
That is absolutely haunting. The idea that a famine in 1944 could reach out and touch a child born in the 1990s is something that sounds more like science fiction than biology. I mean, we are taught in school that our DNA is this blueprint, right? It is the master plan. You get half from mom, half from dad, and that is it. That is your lot in life. If your dad had blue eyes, you might get the gene for blue eyes. We never really considered that what your dad ate for breakfast, or whether your grandmother was stressed out, could fundamentally alter how that blueprint is read. It challenges everything we thought we knew about inheritance. I always assumed that unless there was a mutation, like a random error in the copying process caused by radiation or a chemical, the DNA remained static. But you were saying that the environment, starvation, stress, maybe even toxins can leave a mark that is not a mutation but is still passed on. It is like someone scribbling notes in the margins of the instruction manual. And if that is true, it changes the way we have to look at everything from medicine to sociology. It means we are not just isolated individuals. We are carrying the history of our ancestors in a very literal, biological way. So, this book, The Epigenetics Revolution by Nessa Carey, is essentially claiming that we have missed half the picture for the last century.
Exactly. You have hit on the perfect analogy there. Think of it this way. DNA is the script. Let us say it is the script for Shakespeare's Romeo and Juliet. You can hand that same script to two different directors. One director is Baz Luhrmann, who made that flashy, modern version with Leonardo DiCaprio in the 1990s. The other director is George Cukor, who made a very traditional, classic version in the 1930s. The words on the page are identical. Romeo, Romeo, wherefore art thou Romeo? The text does not change. But the outcome? The movie you watch? It is completely different. One is loud, chaotic, and modern. The other is restrained and classical. Epigenetics is the director. It is the set of instructions that tells the cell how times to read the script. It says, okay, this scene, this gene needs to be shouted. Or, this scene needs to be whispered. Or, cut this scene entirely. We do not need it. Without the director, the script is just paper. Without epigenetics, DNA is just a long, inert molecule. And this explains the fundamental mystery of life that we often overlook because it is so obvious. Differentiation. You have 50 trillion cells in your body. A cell in your retina and a cell in your kidney have the exact same DNA. They have the same script. So why does your kidney not grow eyelashes? Why does your eye not try to filter urine? It is because the epigenetic director has crossed out the eye genes in the kidney cell and highlighted the kidney genes. It is the ultimate form of crowd control for your genes.
That is a brilliant way to visualize it. The kidney cell has the script for the eye, but the pages are glued together. It cannot read them. But this brings up a massive question. If the cells have simply silenced the unwanted genes, is that silence permanent? I mean, for a long time, we thought it was a one-way street. You start as a fertilized egg, a zygote, which is like a blank slate, able to become anything. Then you specialize. You become a skin cell or a liver cell. We used to think that was it. You could not go back. You could not turn a skin cell back into a whole person. But the book talks about this incredible character, John Gurdon, and his ugly toads. He basically proved everyone wrong, didn't he? He took a nucleus from a fully grown, specialized intestine cell of a toad and put it into an empty egg. And it didn't just make more intestine cells, it made a whole new toad. That blew my mind. It means the notes in the margin, the epigenetic instructions, can be erased. The script is still pristine underneath. It is like realizing you wrote your notes in pencil, not permanent marker. That must have been a massive shock to the scientific community when he did that.
It was revolutionary. John Gurdon showed that the genome retains all its potential, even in an old, specialized cell. He basically invented cloning decades before Dolly the sheep came along. And to understand why this matters, we have to talk about a visual metaphor that is famous in biology, Waddington's epigenetic landscape. Imagine a marble at the top of a hill. The hill slopes down and branches into different valleys. The marble is the embryonic stem cell. It has potential. It can go anywhere. As gravity pulls it down, it has to choose a path. Does it roll left into the muscle valley or right into the nerve valley? Once it rolls down into a deep valley, it is stuck there. A nerve cell stays a nerve cell. It cannot just jump over the mountain to become a muscle cell. That is how we viewed development for decades. Gravity only goes one way. But Gurdon, and later Shinya Yamanaka, showed that if you push that marble hard enough, if you use the right tools, you can actually push it back up the hill. You can take a skin cell, which is sitting at the bottom of the valley, and shove it all the way back up to the top, turning it back into a stem cell that can become anything. This is what we call reprogramming. It is not magic. It is stripping away the epigenetic markers that locked the cell into its identity.
And that leads us to Shinya Yamanaka, the time traveler of biology. The book describes his work as almost impossibly optimistic. I mean, think about the complexity of a cell. Thousands of genes interacting. And Yamanaka asked, can I take a grown-up skin cell and turn it back into an embryo-like state? Most people thought you would need hundreds of factors to do that. It is like trying to turn a baked cake back into flour, eggs, and sugar. But he found out you only needed 4. Just 4 genes. OCT4, SOX2, KELF4, and c-MYC. That is it. If you force a skin cell to express these 4 factors, it forgets it is a skin cell. It forgets its history. It becomes an induced pluripotent stem cell, or iPS cell. It is like he found the factory reset code for life. The implications for medicine are staggering. If you have heart disease, we could potentially take some skin from your arm, reset it, turn it into fresh heart muscle cells, and patch up your heart. No rejection, because it is your own DNA. It is the ultimate repair kit. But the book also mentions that this process is incredibly inefficient compared to nature. Nature resets the sperm and egg almost instantly upon fertilization. In the lab, it takes weeks and works on only a tiny fraction of cells. We are still clumsy toddlers compared to nature's elegance.
It is true. Nature is the master. But let us look at another natural experiment that confuses people. Identical twins. If genetics were the only thing that mattered, identical twins would be, well, identical in every way. They have the exact same DNA sequence. They are clones of each other. But we see time and time again that they are not the same. One might develop schizophrenia, and the other does not. One might get breast cancer, the other stays healthy. One might be gay, the other straight. If the blueprint is the same, why is the house different? The book calls this the discordance. For schizophrenia, if one identical twin has it, the chance the other has it is only about 50%. That is high, sure, but it is not 100%. Where is the missing 50%? It is in the epigenome. As twins age, their lives diverge. One might smoke, the other exercises. One might suffer a traumatic breakup, the other marries happily. These experiences cause their epigenetic markers to drift apart. A study by Manel Esteller showed this beautifully. He looked at the chromosomes of young twins and they lit up, almost identical. Then he looked at old twins who had lived apart. Their chromosomes looked completely different in terms of epigenetic marks. They had become different people, biologically, despite having the same DNA. It is proof that we are not just our genes. We are the accumulation of our experiences layered on top of our genes.
Okay, so we have established that this layer exists. It explains differentiation, it explains stem cells, and it explains why twins are different. But what is it physically? We cannot just keep calling it a ghost or a director. We need to get under the hood. The book describes two main mechanisms, and the first one is DNA methylation. I love the analogy used here. DNA is a long zipper, right? With the teeth being the letters A, C, G, and T. Methylation is like sticking a grape on one of the teeth, specifically the C tooth, cytosine. Chemically, it is a tiny little carbon group. It is minuscule. Sticking a grape on a tennis ball is the scale. But if you stick that grape on the zipper, the zip gets stuck. The machinery that reads the DNA comes zooming along, hits that methyl group, and falls off. It cannot read the gene. So methylation equals silence. It turns the gene off. And the craziest part is that this grape gets copied. When the cell divides, an enzyme comes along, sees the grape on the old strand, and immediately sticks a new grape on the new strand. That is how the memory is passed on. That is how your liver cells remember to be liver cells for 70 years. They keep copying the silence.
Right. Methylation is the heavy-duty lock. It is the do not open sign. But there is a second, more nuanced mechanism involving proteins called histones. If DNA is the thread, histones are the spools that the thread wraps around. If you wrap the thread really, really tight, you cannot read the script on the inside. If you loosen it up, the genes become accessible. This is where the histone code comes in. It is incredibly complex. The book compares it to decorating a Christmas tree. You have these histone tails sticking out like branches. You can hang different chemical decorations on them: acetylation, methylation, phosphorylation. Each decoration means something different. Some say unwind a bit. Others say pack it tight. It is like a dimmer switch rather than an on-off switch. And this is where the environment really gets to play. Things like stress hormones, diet, or drugs can change these decorations rapidly. Acetylation usually opens things up. It turns the gene on. Deacetylation shuts it down. This system is much more flexible than DNA methylation. It is dynamic. It allows your cells to respond to the world in real time. It is the reason why, if you start working out, your muscle cells change their gene expression patterns to build more protein. The DNA did not change, but the histones opened up the grow muscle genes because of the signal from your exercise.
So we have the zipper and the spool histones. These are the tools, but let us see them in action. The book talks about this incredible experiment with Agouti mice. This one really stuck with me because it is so visual. You have these two mice. They are genetically identical clones, but one is fat and has yellow fur. The other is thin and brown. The fat yellow mouse is prone to diabetes and cancer. The thin brown mouse is healthy. The difference? It is all down to one gene, the Agouti gene. In the yellow mouse, this gene is stuck in the on position because of a lack of methylation. It is like a car alarm that will not stop blaring. It makes the mouse yellow and ravenously hungry. In the brown mouse, the gene is methylated. It is silenced. The alarm is off. But here's the kicker. You can change the outcome by changing the mother's diet. If you feed the pregnant mother a diet rich in methyl donors, things like folic acid, vitamin B12, basically leafy greens and garlic, she produces healthy brown babies. If she eats a poor diet, she has fat yellow babies. The food the mother ate directly provided the raw materials, the grapes, to silence the bad gene in her babies. It is the most direct proof of you are what your mother ate I have ever heard.
It is undeniable proof. And it shows that nature and nurture are not enemies. They are partners. The book uses the analogy of the board game Mousetrap. You know, that crazy Rube Goldberg machine game? You have the boot, the bucket, the stairs, the diving man. Those parts are the genes. They are the hardware. But for the trap to work, you need the ball to start rolling. That ball is the environmental signal. And everything has to be lined up perfectly. If one epigenetic piece is out of place, if the boot gene is methylated when it should be active, the whole trap fails. This brings us to human diseases that are purely epigenetic. The most heartbreaking one mentioned is Rett syndrome. It affects little girls. They are born seemingly healthy, but then they start to regress. They lose their speech. They lose control of their hands. It turns out they have a mutation in a gene called MECP2. This gene is a reader. Its job is to spot the methylation grapes on the DNA and say, aha, this gene is off. I will make sure it stays off. Without a working MECP2, the brain cells cannot read the silence. Genes that should be quiet start shouting. It is chaos in the library. The brain cannot develop properly because the instructions are being misread. But, and this is the hopeful part, Adrian Bird's lab showed that if you fix the MECP2 gene in mice, even after they have developed symptoms, they recover. It suggests that the brain is not broken forever. It is just waiting for the correct software update.
That is incredible. It implies that conditions we thought were permanent brain damage might actually be reversible. But I want to go back to the inheritance part, the grandmother effect. We talked about the Dutch hunger winter, but there is this lingering question, is it really epigenetic inheritance, or is it something else? The book introduces a skepticism here. When a woman is pregnant with a daughter, that daughter already has eggs forming in her ovaries. So you have 3 generations in one body, the mother, the fetus, and the germ cells, grandchildren. So if the grandmother is starving, she is directly affecting the eggs that will become the grandchildren. That is not necessarily inheritance of a trait, it is direct exposure. But what about men? Men do not carry 3 generations. Sperm are made fresh. So if a father's experience affects his children, that is a cleaner proof of epigenetic inheritance. And the book takes us to this tiny place in Sweden called Överkalix. They have these meticulous records of harvests, feast and famine. And they found something that makes you want to put down that donut immediately. If a grandfather had a glut of food, if he stuffed his face during his slow growth period just before puberty, his grandsons were 4 times more likely to die of diabetes. 4 times! The grandfather's gluttony killed the grandson. That is biblical. The sins of the fathers, literally.
It is terrifying, isn't it? It suggests that the sperm carries more than just DNA. It carries a memory of the father's metabolic state. It is sending a signal to the next generation saying, hey, the environment is full of food. Program your body to hoard sugar. But in a modern world where food is everywhere, that programming backfires and causes diabetes. This brings us to a massive scientific controversy, Lamarck versus Darwin. Jean-Baptiste Lamarck was this guy before Darwin who thought that if a giraffe stretched its neck to reach leaves, its babies would be born with longer necks. He believed in the inheritance of acquired characteristics. Darwin came along and said, no, it is random mutation and natural selection. And poor Lamarck became the laughingstock of biology for 200 years. But epigenetics is basically saying, hold on, Lamarck was not entirely crazy. You cannot grow a longer neck, but you can pass on a metabolic setting. You can pass on a stress response. It is a soft inheritance. It is not permanent, it can fade over generations, but it is real. The Agouti mouse proves it. The Överkalix study proves it. We are looking at a hybrid evolution where Darwin is the main driver, but Lamarck is sitting in the passenger seat giving directions.
It really makes you rethink personal responsibility. I mean, if I smoke, I am not just hurting my lungs, I might be flipping epigenetic switches in my future children. It adds a heavy weight to our lifestyle choices. But let us pivot to something more hopeful. Medicine. If these epigenetic marks are reversible, like we saw with the iPS cells and the Rett syndrome mice, can we use that to cure disease? The book talks about cancer. Cancer is often a disease of gene regulation gone wrong. Tumor suppressor genes get silenced by methylation. They get locked up. The book mentions drugs that are already being used, like azacitidine. These are demethylating agents. They basically strip the grapes off the DNA zipper. They unlock the good genes so they can get back to work fighting the cancer. It is not just theoretical, it is happening now. And then there is the potential for regenerative medicine. If we can master the Yamanaka factors, we could grow new organs. We could treat diabetes by making new insulin-producing cells from a patient's own skin. We are standing on the edge of a new era where we stop just treating symptoms and start editing the software of the cell itself.
We absolutely are. But the book also ends with a note of caution. This system is insanely complex. We talked about the Christmas tree histones. Well, imagine trying to adjust one ornament without knocking off 10 others. Epigenetic drugs can be blunt instruments. If you strip methylation from the whole genome, you might wake up the good genes, but you might also wake up some ancient viruses hidden in our DNA or turn on genes that should stay asleep. It is a balancing act, and we are still learning the rules. But the main takeaway from the Epigenetics Revolution is awe. Just sheer awe at the complexity of life. We are not just machines following a blueprint. We are dynamic, responsive entities. Our bodies are constantly conversing with our environment. Every meal, every run, every stress, every joy, it is all being recorded in the molecular marginalia of our cells. And astonishingly, some of those notes might be read by our children. It is a beautiful, if somewhat daunting, realization. It means we have more agency than we thought, but also more responsibility. We are the guardians of our own genome, and partially, the guardians of the generations to come.
Well said. It really does change how you view your place in the world. You are not just a passive carrier of DNA. You are an active participant in your biological destiny. The work of scientists like Gurdon, Yamanaka, Bird, and all the others mentioned in this book has opened a door that can never be closed. We have moved from the century of the gene to the century of the epigenome. And who knows what we will find next. Maybe one day we will be able to precisely edit these epigenetic marks to eliminate mental illness, or extend human lifespan, or reverse the effects of trauma. It is a frontier as vast as space itself, but it is happening right inside our own nuclei. Thank you for joining us on this deep dive into the epigenetics revolution. It has certainly rewritten my understanding of what it means to be human. If you enjoyed this, maybe go eat some broccoli. Your future grandson might thank you for it one day.